morning students, we will continue the next topic of lipid metabolism, competition number 4.2, and today's topic is formation, fate of ketone bodies, significance, regulation and associated disorders like ketosis. First about the ketone bodies. So there are three ketone bodies found in our body, namely acetoacetic acid, beta hydroxybutyric acid and acetone. First we will see the formation of ketone bodies, which is called as ketogenesis. It is a synthesis of ketone bodies. Site ketogenesis occurs in the mitochondria of the liver cell, and substrate required for synthesis of ketone body is acetyl CoA. Now this acetyl CoA is available for ketone body synthesis from beta oxidation of fatty acid, which is also occurring in the same compartment, that is mitochondria of liver cell. Ketogenesis takes place in the fasting state of the body, and the hormones responsible for ketogenesis are glucagon and epinephrine. So this is the ketogenesis pathway. It is a very simple pathway. It occurs in 3 steps. First, the 2 molecules of acetyl CoA with 2 carbon atom each condenses to form acetoacetyl CoA with the help of enzyme acetoacetyl CoA synthase. The 4 carbon acetoacetyl CoA then combines with 1 more molecule of acetyl CoA to form beta hydroxy beta methyl glutaryl CoA. In this step, one coenzyme A molecule is released and enzyme is HMG CoA synthase. The long form of HMG is beta hydroxy beta methyl glutaryl CoA. In the third step, this HMG CoA undergoes lysis with the help of enzyme HMG CoA lyase and gives 1 molecule of acetyl CoA and 1 molecule of acetoacetate or acetoacetic acid with 4 carbon. So 6 carbon HMG CoA is broken down into 2 carbon acetyl CoA and 4 carbon acetoacetate or acetoacetic acid. Now this acetoacetate is also called as primary ketone bodies. Now this acetoacetate undergoes spontaneous decarboxylation to form another ketone body, that is acetone, or this acetoacetate may undergoes reduction to form a third type of ketone body, that is beta hydroxy butyric acid with 4 carbon. Here 2 hydrogen atoms are added, which are supplied by NADH. The enzyme required for this step is beta hydroxybutyrate dehydrogenase. So this reaction, this enzyme catalyzes the reverse reaction in which it converts the beta hydroxybutyrate into acetoacetate. At that time, this enzyme removes the 2 hydrogen atom from beta hydroxybutyrate, and hence it is named as beta hydroxybutyrate dehydrogenase. As this enzyme catalyzes the reversal reaction, we will consider the same name for conversion of acetoacetate to beta hydroxybutyrate. So this is about the ketogenesis cycle. After ketogenesis, we will see ketolysis. Ketolysis means breakdown of ketone bodies. Again, here site is mitochondria and cytoplasm of all cells except liver, but mainly ketolysis occurs in the mitochondrial compartment of all body cells except liver. Here it occurs in fasting state of the body. Again, the hormones responsible for ketolysis are glucagon and epinephrine. Now we will see the ketolysis. First we will see ketolysis in mitochondria. In the mitochondria, the ketone body beta hydroxybutyrate undergoes oxidation by removal of 2 hydrogen atoms, which are accepted by NAD to form NADH, and it gives acetoacetate, which is another ketone body. The enzyme required for this step is the same enzyme of ketogenesis pathway, that is beta hydroxybutyrate dehydrogenase. Now this acetoacetate or acetoacetic acid then combines with succinyl CoA and get converted into acetoacetyl CoA. Here coenzyme A from succinyl CoA is added to acetoacetate with the help of enzyme thiophorase, which is present in the mitochondrial compartment of all cells of the body, and succinyl CoA is converted into succinic acid. Now this acetoacetyl CoA then undergoes lysis with the help of enzyme thiolase. Here one coenzyme A molecule is added, and 4-carbon acetoacetyl CoA is converted into 2 molecules of 2-carbon acetyl CoA. So in this way the end product of ketolysis is acetyl CoA. Substrate for ketone body synthesis is acetyl CoA, as well as end product of ketone breakdown is also acetyl CoA. Now we see ketolysis in cytoplasm. Here the first step is same, in which beta hydroxybutyric acid with 4-carbon is converted into acetoacetate by oxidation. 2 hydrogen atoms are removed, which are accepted by NAD to form NADH. The enzyme is also same, that is beta hydroxybutyrate dehydrogenase. Now in the second step, instead of enzyme thiophorase, the enzyme acetoacetyl CoA synthase is there in the cytoplasma. This enzyme add free coenzyme A molecule to acetoacetate to form acetoacetyl CoA. Here ATP is required. In the mitochondrial step, ATP is not utilized. So this pathway is minor pathway for ketolysis. The pathway of ketolysis in the mitochondria is the major pathway. So acetoacetyl CoA is formed with 4-carbon, which undergo lysis with the help of enzyme thiolase to form 2 molecules of acetyl CoA. This step is also similar to the step in mitochondria. So this is the comparison between the ketolysis in mitochondria on left side and ketolysis in cytoplasma on the right side. Here first step is same in both cases. Third step is also same. The only difference is the enzyme. In mitochondria the enzyme thiophorase is there. In cytoplasmic compartment the enzyme is acetoacetyl CoA synthase is there. Here second difference is coenzyme A is added to the acetoacetic acid in mitochondrial compartment from succinyl CoA. Here free coenzyme A is added. Therefore in cytoplasmic compartment, to add this coenzyme A into the acetoacetate to form acetoacetyl CoA, energy is required, which is supplied by ATP, and in mitochondrial compartment ATP is not required. So ketolysis occurs in the mitochondria is the major pathway of ketolysis. After completing the synthesis and breakdown of ketone bodies, we will see the regulation of ketone body metabolism. First, hormonal regulation, or it is the only regulation for ketone body metabolism. Glucagon and epinephrine stimulates the synthesis as well as lysis of ketone body, whereas insulin inhibit the ketogenesis and ketolysis process. These hormones regulate the ketone body metabolism at 3 different levels. At level 1, the lipolysis in adipose tissue is regulated by these hormones. So all of us know that in adipose tissues triglycerides are stored. In fasting state, the enzyme hormones glucagon and epinephrine stimulate the enzyme hormone sensitive lipase. Now this enzyme hormone sensitive lipase acts on triglycerides, break the triglycerides into glycerol and fatty acids, and these fatty acids are then transported to the liver for beta oxidation, and the end product of beta oxidation is acetyl CoA, and these acetyl CoA are used for ketone body synthesis. So this is the level 1 where ketone body metabolism is regulated by hormone. Insulin inhibit the enzyme hormone sensitive lipase, so it stops the lipolysis in adipose tissue. Regulation at the entry of fatty acid into the mitochondria, that is level 2 regulation. In beta oxidation pathway, I have told you that the fatty acids from adipose tissue first enters into the cytoplasm of the liver cell. In the cytoplasm, these fatty acids are activated and then they get transported into the mitochondrial compartment for beta oxidation. For transport of fatty acid from cytoplasmic compartment to the mitochondrial compartment, they require enzyme called as carnitine acyl transferase 1. Now this carnitine acyl transferase 1 is under the control of malonyl CoA. Malonyl CoA is the allosteric inhibitor of carnitine acyl transferase 1. Now this insulin increases the synthesis of malonyl CoA. This malonyl CoA allosterically inhibit the carnitine acyl transferase 1 so that the fatty acid does not enter into the mitochondria and not available for beta oxidation. In contrast, glucagon and epinephrine in the fasting state inhibits the synthesis of malonyl CoA. Therefore, the activity of carnitine acyl transferase 1 increases and it will transport more and more fatty acid from cytoplasmic compartment to the mitochondrial compartment for beta oxidation. So this is the regulation of ketone body metabolism at level 2. In the last level, the oxidation of acetyl CoA is regulated. Normally, all of us know that in the mitochondria, the acetyl CoA combines with oxaloacetate and enters into the TCA cycle to form citric acid. But in the fasting state, there is increased glucagon to insulin ratio. Therefore, the oxaloacetate which is available in the mitochondria of the liver cells is diverted for gluconeogenesis pathway. That means synthesis of glucose, because in the fasting state we have to supply, body has to supply glucose to the brain and RBC. So glucose is synthesized from non-carbohydrate compounds like oxaloacetate, pyruvic acid, amino acid. So in fasting state, due to increased glucagon to insulin ratio, divert the oxaloacetate to gluconeogenesis pathway for synthesis of glucose. Hence, acetyl CoA, less oxaloacetate is available to combine with acetyl CoA to enter into the TCA cycle. So in this case, two things had happened. First, in the liver mitochondria, the concentration of acetyl CoA is increased because of beta oxidation pathway. And on second thing is the level of oxaloacetate is decreased. Hence, these acetyl CoA are not fully utilized in TCA cycle because of non-availability of oxaloacetate or decreased availability of oxaloacetate. Hence, the excess of acetyl CoA which are present in the liver mitochondria are diverted for ketone body synthesis. So, in this way, at three different levels, the ketone body synthesis is regulated. After completing the regulation of ketone bodies, we will study the significance of ketone bodies. Why ketone bodies are synthesized? For that purpose, you should know something about post-meal state and fasting state. What is post-meal state? Post-meal state means it is a state of the body after meal. In post-meal state, glucose is available. So this glucose enter into all cells of the body. When glucose enters into the cells, it undergoes glycolysis in the cytoplasm to form pyruvate. That is glycolysis pathway. Then this pyruvate enter into the mitochondria and forms acetyl CoA. Now this acetyl CoA is used for TCA cycle. In TCA cycle, two substrates are required. One is oxaloacetate and another is acetyl CoA. If you remember the TCA cycle, at the end of TCA cycle, oxaloacetate is regenerated. That means only oxaloacetate is utilized, only acetyl CoA is utilized in the TCA cycle, whereas oxaloacetate is regenerated. So if we want to run a TCA cycle, what is the requirement? The requirement is of acetyl CoA, not oxaloacetate. So in post-meal state, this acetyl CoA comes from glucose through glycolysis pyruvate, and pyruvate is converted to acetyl CoA in mitochondria with the help of pyruvate dehydrogenase enzyme. So all cells of the body will get acetyl CoA from glucose in the post-meal state. But in fasting state, the situation is different. In fasting state, glucose is not available. Therefore, triglycerides present in the adipose tissue undergoes lipolysis to form fatty acids. These fatty acids are transported to the liver for beta oxidation. In the liver mitochondria, beta oxidation occurs and acetyl CoA are formed in large quantity in liver mitochondria only. So this acetyl CoA, which is formed in the liver mitochondria, then we have to transport this acetyl CoA to all peripheral tissues to run the TCA cycle in that cells. But problem is, this acetyl CoA cannot crosses the mitochondrial membrane, or mitochondrial membrane does not permit the acetyl CoA to cross the membrane. Therefore, to overcome this difficulty, the body will synthesize the ketone bodies in the mitochondrial membrane. Why ketone bodies? Because ketone bodies are readily soluble in water. Therefore, they are easily transported into the blood. The next and most important property of ketone bodies is they can easily cross the mitochondrial membrane. Hence, the body will synthesize the ketone bodies from the acetyl CoA which are available in the liver mitochondria, and after converting the acetyl CoA into ketone bodies, then ketone bodies passes through the mitochondrial membrane, reaches the peripheral tissue. In the peripheral tissues, these ketone bodies are taken up by the mitochondrial cells, the mitochondria of the cells. These ketone bodies are broken down and acetyl CoA is regenerated, and that acetyl CoA is used to run the TCA cycle. So this is the significance of ketone bodies. We have studied the ketolysis of only two ketone bodies, that is acetoacetate and beta hydroxybutyrate. Now what about acetone? Acetone is a volatile ketone body. That means it gets evaporated at body temperature. Therefore, it can be easily excreted through the breath. If a person breathe out acetone, then the smell of his breath is fruity odor. Similarly, acetone are water soluble, so they get excreted into the urine, and a very small part of acetone is converted into acetoacetate. That means acetone has no metabolic significance as far as its fate is concerned. It is just excreted from the body either through the breath or through the urine. Now coming to the disorders of ketone body metabolism, which is called as ketosis. Ketosis is a combination of three conditions: ketonemia, ketonuria and fruity odor to the breath. Ketonemia means increase in the ketone bodies in the blood. Ketonuria means excretion of ketone bodies in the urine. And these two conditions along with fruity odor to the breath due to presence of acetone in the breath, when all these three conditions combines, then it is called as ketosis. Causes of ketosis are starvation, severe diabetes mellitus and hyperemesis, means excessive vomiting. Out of these three, first two causes are very important: starvation and diabetes mellitus, because they are seen very commonly in the patients. Now, what is the effect of ketosis? Now, ketosis causes deficiency of bicarbonate. Why? Ketone bodies are acidic in nature, means acetoacetic acid, beta hydroxybutyric acid. So they are acidic in nature. They releases H plus ion in the blood. To neutralize that H plus ion, the bicarbonates which are present in the blood are utilized. Therefore, there is deficiency of bicarbonate. When excessive production of ketone bodies is there, then they releases large amount of H plus ion in the blood, which decreases the pH of the blood to a small extent, causing acidosis. Whenever there is acidosis, it stimulates the respiratory centers in the medulla oblongata, because these centers are highly sensitive to H plus ions. When the respiration is stimulated, there is increase in rate and depth of respiration. Therefore, more and more carbon dioxide is removed from the body. When carbon dioxide level in the blood or PCO2 decreases, then to maintain that PCO2 level, carbonic acid H2CO3 is broken down into carbon dioxide and water. So to maintain the PCO2, H2CO3 is broken down. Therefore, there is decrease in carbonic acid concentration of the body. Next is fruity odor to the breath due to presence of volatile ketone body, that is acetone. Next condition is diuresis. Why diuresis? Ketone bodies, I have told you, they are readily soluble in water. They get readily filtered through the glomerulus and enter into the tubules or in the filtrate of present in the tubules. Due to presence of ketone bodies in the filtrate of the tubules, there is increase in the osmolarity of that filtered urine or filtrate. And when the osmolarity of the filtrate increases, that filter will drag the water from the peripheral blood vessels, that is vasa recta and tubular cells, into the tubule by osmosis. So large amount of water enter into the tubules, and when the water enters into the tubules, it is excreted into the urine, leading to diuresis. Diuresis means excretion of large amount of urine. When there is loss of more water through urine, it leads to dehydration. Next is hyponatremia. Hyponatremia means decrease in the sodium level of the body. Why sodium level decreases? Because I have told you ketone bodies are acidic in nature. They releases H plus ion in the blood. Therefore, ketone bodies will acquire a negative charge. When they acquire a negative charge, they readily combines with a positive sodium ion. So ketone bodies combines with sodium ion. When the ketone bodies after filtration get excreted into the urine, sodium is also excreted along with the ketone bodies. So there is loss of sodium from the body. And if the condition becomes more severe, it affects the brain and patient often goes in coma. So these are the effects of ketosis. Now, how will you diagnose ketosis? The most important test is Rothera's test on urine sample. If ketone bodies are present, then Rothera's test is positive. This Rothera's test is given by all three ketone bodies. Another test on urine is Gerhardt's test. It is also a test for presence of or detection of ketone bodies in the urine. Next test is measurement of ketone bodies in the blood. Normally, less than 1 milligram per deciliter of ketone bodies are present in the blood. But in case of ketosis, the level of ketone bodies in the blood increases. That condition we name as ketonemia. So if you measure the level of ketone bodies in the blood, you will get high levels. Next, measurement of blood glucose level. In the causes of ketosis, I have told you there are two important causes of ketosis. One is starvation and another is diabetic ketoacidosis. If you want to differentiate between these two, then this blood glucose level is very important. In case of starvation, blood glucose level is low, that is hypoglycemia, and in case of severe diabetes mellitus, the blood glucose level is very high, hyperglycemia is there. So blood glucose level helps in the distinguishing the cause of ketosis. And last test is Benedict test on urine sample. It detects the presence of glucose in the urine. In case of diabetes mellitus, when blood glucose level is high, then glucose is excreted into the urine, and which is detected by the Benedict test. Lastly, management of ketosis. So in management, we have to give glucose if the cause is starvation. Insulin is given to the patient if the cause of ketosis is diabetes mellitus. But you have to take precaution before giving insulin. Insulin is only given only after the confirmation of blood glucose level. Next, bicarbonate deficiency is there, so we have to supply bicarbonate. Dehydration is treated by fluid replacement. Hyponatremia is treated by giving electrolytes. So in this way, the ketosis is managed. Frequently, a case is asked on this topic. So one of these cases given here. A 45-year-old man in coma is admitted to the hospital. Generally, the ketosis patient comes to the hospital with coma. His breath have fruity odor and blood sugar level is 550 milligram per deciliter. So this is the case given. Now questions on this case are: what is the most probable diagnosis? Blood glucose level is high, patient is in coma, and fruity odor of the breath. So these 3 points indicates that the patient is suffering from diabetic ketoacidosis. Now what are the biochemical changes seen in these patients? The biochemical changes just we have studied. These are biochemical changes: deficiency of bicarbonate, decrease pH, that is acidosis, decrease PCO2 level, dehydration is there, decreased sodium level is there. So these are the biochemical findings seen in these patients. How will you diagnose? Just now I have shown you the various tests in blood as well as in the urine for diagnosis. Rothera's test and Gerhardt's test in urine for detection of ketone bodies. Estimation of blood ketone body level. Estimation of blood glucose level helps in the diagnosis of the cause. and sometimes Benedict test in urine is also used for detection of glucose in urine. Now how will you manage the patient? That is also I have shown. Management includes glucose if the cause is starvation, insulin if the cause is diabetes mellitus, cause of ketosis is diabetes mellitus, but precaution is we have to confirm the blood glucose level, then only we have to give insulin. Then bicarbonate supplements, fluid replacement therapy and electrolyte balance. So this is the management of the patient, and this is about ketone body synthesis, its breakdown, regulation, significance and disorder called ketosis. Thank you very much.